Today I'm talking to Dr. Sebastian Fuchs, who's a new Crim Fellow with amfAR. He's working at the University of Miami, and we're looking forward to getting to know more about him and his projects. Thanks for joining us, Sebastian.
Thank you for having me here.
So let's start with talking about the project you applied to amfAR for. What are you going to be working on for the next couple of years?
It's basically an alternative approach to prevent and treat HIV infection. Currently, and over the last decades, the main focus has been vaccine development and traditional vaccines. Now, lately, also vaccines based on mRNA, lipid nanoparticles, and traditional vaccines have not been that successful. About 7 efficacy trials have been conducted so far, and none of them showed any efficacy at all. I've been working for the last, I would say, since 2009 on a gene therapeutic approach, which is an alternative approach, and it utilizes a viral vector called AAV, and it's a non-pathogenic virus. And it's actually a very elegant solution because when we talk about a vaccine, we are delivering to an individual a pathogen or an inactivated pathogen or part of it, protein, and we hope to basically induce a protective immune response against the pathogenic or the virulent form. However, with HIV, it's incredibly difficult. One has to imagine one of the biggest problems with HIV is its sequence diversity. And one has to imagine that.
And when you say sequence diversity, you mean that it has a lot of genetic mutations?
Correct. The mutation rate is enormous. And one has to imagine that the genetic sequence diversity of all virus particles of HIV within one infected individual equals or exceeds the sequence diversity of all flu strains, viruses in all people in the entire world in a given year. I mean, that actually highlights what a problem HIV is. Instead of hoping of an immune response by a vaccine, we're delivering the immune response. And that using this viral vector, AAV. And so what AAV does is it's basically just a non-pathogenic, non-multiplying, non-replicating virus is injected into a non-dividing or slowly dividing tissue such as muscle, skeletal muscle. And the muscle then produces a well-characterized or several well-characterized antibodies that are specific against HIV, which are very potent and which are also very broad. And a lot of studies, including our own, have shown that it can basically suppress virus in infected, in our case, animals. It has also shown very potent antiviral effect in human clinical trials for the antibodies when infused. So the hope is here to basically use AAV to establish a protective barrier against HIV viral acquisition. And in a therapeutic scenario, it is hoped to basically suppress viremia the same way as antiretroviral therapy would do. But the beauty is, as my mentor Ron would say, one shot and you're good for life.
Yeah.
And that's the goal.
In a nutshell, I think, people might be a little bit surprised, but in a nutshell, you're using one virus, one that does not cause disease, to deliver the information into a person's arm muscle that
correct.
that person can then make antibodies for the rest of their life. And I think, people are kind of familiar with antibodies now because we've heard so much about them with COVID. But, antibodies can be effective against all kinds of viruses. And your hope is that you can get a person's arm muscle to make antibodies against HIV so that that could prevent them from getting HIV in the first place, or if they have HIV, it could treat them.
Correct. That is correct. I think the first step would be to use it in HIV-infected individuals. First and foremost, probably in patients that are almost terminally ill, and to give them the option to receive this gene therapy approach with the hope that they would basically really benefit from their treatment. And then,
it might be good for people who are resistant to existing antiretroviral therapy, for example.
That is correct. And we should also not forget that antiretroviral therapy, known as ART, does suppress viremia, but of course there are also, escape mutants, and there are toxicities associated with the drugs, and also the burden to have to take those drugs daily. Although recently there have been drugs developed that are long lasting after a single injection for 1 or 2 months, but still it requires frequent administration.
I want to ask you, do you think there's a chance that your antibodies and the way that you deliver them might play a role in curing HIV?
Yes, I do believe that. And I mean, it's also backed up by data that antibodies do exert not only antiviral effect against the virus particle, but also against virally infected cells. And it's true that there are compartments in the human body where the virus hides, and those are not necessarily accessible to antibodies. But we have shown in our publication that antibodies have effector functions and that they can actually kill virally infected cells and reducing the viral reservoir much faster than ART would do.
That's the beauty, I think, about antibodies, right? You've got infected cells that produce viruses, and the antibodies can attack and kill these viruses, but they can even attack and kill the cells that are essentially the factories for those viruses.
Correct.
But what made you get interested in HIV in the first place?
To be honest, I came to it by accident. I actually I did my master's in plant biochemistry and plant molecular biology, and I didn't want to just write my thesis on one of those topics. So I was looking, and I one of my minor subjects was immunology. So, and I was fascinated by antibodies, and I learned all the techniques from the plant field as well. So I went to a company in California called Planet Biotechnology back then, and I was allowed to do my master thesis project there. And my task was to produce human antibodies in tobacco plants, with the goal of producing a lot of protein material to be used for infusion purposes. And then I applied for a Ph.D. program that was established between the University of Erlangen in Germany and the New England Primate Research Center of Harvard Medical School. I was interviewed there by Ron DeRojas and other faculty members who flew to Germany, and I was selected. And so basically my interest in antibodies and a publication that was just published back then in April 2009 by Phil Johnson using AAV to protect against immunodeficiency virus infection. that actually sparked my interest.
Well, so you mentioned that you were in Germany, and of course, Germany has a long history of producing a lot of scientists. But what made you interested in becoming a scientist?
I think science has been, my family is full of artists. So I was a musician and so on. And I was kind of like, I needed something more grounding, I would say. And science is very grounding. It's not about it, it's just about facts. And you have to, and of course, I I didn't realize when I was developed maybe that desire to go into science that it's tough and it's about research and discovering and failure and so on. But I think curiosity and finding something out that no one has ever found out before.
Well, we wish you all the best with that and look forward to hearing great things come from you. Thanks for talking to us today.
Thank you very much, Rubina.